Welcome to the InnoGuide Podcast, where we share the works of authors and mentors to guide innovation. In this episode, number 40, we'll be sharing A Brief History of Intelligence, Part 1 by Max Bennett. Introduction. Let me take you back to September 1962, a time when the world was buzzing with the space race, the Cuban Missile Crisis, and the rollout of the polio vaccine. Amidst all this, something quietly revolutionary happened. Humanity predicted the future. No, not through science or politics, but through the cartoon The Jetsons. This colorful sitcom about a family living 100 years in the future wasn't just entertainment, it was a vision of what life could look like, complete with futuristic technologies. And you know what? They got a lot right. Video calls, flat-screen TVs, cell phones, 3D printing, and smartwatches, all of these were wild fantasies in 1962, but are now everyday realities. But there's one prediction from the Jetsons that remains stubbornly out of reach: Rosie the robot. Rosie wasn't just a machine. She was a fully autonomous human-like robot who could cook, clean, teach, and even empathize. She was part of the family, capable of understanding emotions, resolving conflicts, and even falling in love. Yet, here we are in 2025, and we still can't build a robot that can reliably load a dishwasher, let alone one with Rosemary's emotional intelligence. Why? It's not a mechanical problem. It's an intellectual one. The ability to identify objects, handle them delicately, and navigate the complexities of human interaction is far more challenging than we ever imagined. That said, the progress in artificial intelligence, AI, since 1962 has been nothing short of remarkable. AI can now beat humans at chess and Go, diagnose tumors as well as radiologists, and even drive cars, almost. And with the advent of large language models like ChatGPT, AI can write essays, compose poetry, and pass the bar exam better than most lawyers. But does this mean AI truly understands these tasks, or is it just really good at mimicking patterns? For example, does a calculator understand math, or does ChatGPT understand the law? These are the questions that keep us up at night. In 2021, I started exploring these questions while working with GPT-3, the precursor to ChatGPT 4.x that's out now. It was fascinating and frustrating. GPT-3 could write a coherent essay about 18th century potato farming, but couldn't answer a simple question about what you might find in a basement if you looked up. It thought it would see the stars. Why? What aspects of human intelligence does AI capture, and what does it miss? And why do some problems that seem impossible one year become solvable the next? Even with the release of GPT-4 in 2023, which improved on many of GPT-3's shortcomings, we're still far from understanding the full picture. This brings us to the heart of the matter, the gap between human and artificial intelligence. Why can AI dominate at chess but fail at something as simple as loading a dishwasher? The answer lies in the fact that we don't fully understand the thing we're trying to replicate, human intelligence. And to understand it, we might need to look beyond the human brain itself. Nature's clues and the complexity of the brain. When humanity wanted to understand flight, we looked to birds. When George de Mestral invented Velcro, he studied burdock fruits. Nature has always been our guide, so naturally when it comes to understanding intelligence, we turn to the human brain. But here's the problem. The brain is really complicated. It has 86 billion neurons and over 100 trillion connections, all packed into a tangled, ever-changing web. Even if we could map every single connection, we'd still be far from understanding how it all works. Unlike the straightforward electrical signals in a computer, the brain communicates using hundreds of different chemicals, and its connections are constantly rewiring themselves. It's a marvel and a mystery. So if the human brain is too complex to decipher directly, where else can we look? The answer might lie in the past. By studying fossils, genes, and the brains of other animals, we can piece together the evolutionary story of how intelligence came to be. After all, the human brain didn't appear out of nowhere. It's the result of millions of years of evolution, starting with a tiny worm that lived 600 million years ago. This worm's simple brain was the foundation of all the diverse and complex brains we see today, including our own. The evolutionary story of intelligence. To understand the human brain, we need to trace its evolutionary journey. This book is organized around 5 major breakthroughs in brain evolution, each of which added new intellectual abilities to our ancestors. These breakthroughs didn't happen in isolation. They were shaped by the challenges our ancestors faced, from outsmarting predators to surviving environmental disasters. And here's the exciting part. Many of these breakthroughs have parallels in artificial intelligence. By studying how intelligence evolved in nature, we might uncover clues to building better AI. But let's be clear, this isn't about creating a hierarchy of intelligence. Evolution doesn't rank species as better or worse. Every species alive today is equally successful in its own niche. An octopus, for example, is a multitasking genius, while a fish can navigate obstacles at lightning speed. Humans are unique, yes, but not superior. This book focuses on the human story, but it's just one of many stories that could be told. Why I wrote this book? I'm not a neuroscientist or an AI researcher. I'm just someone who became fascinated by the gap between human and artificial intelligence while working in the tech industry. My company, Bluecore, used AI to predict consumer behavior, and I was constantly struck by how much we take human intelligence for granted. Tasks that seem simple to us, like recognizing objects or understanding context, are incredibly difficult for AI. This curiosity led me down a rabbit hole of research, conversations with neuroscientists, and eventually the decision to write this book. My goal is to provide an accessible introduction to how the brain works, why it works the way it does, and how it compares to AI. Along the way, I'll share the evolutionary story of intelligence grounded in both biology and the latest advancements in AI. It's a story of breakthroughs, challenges, and the tantalizing possibility that nature holds the key to building human-like artificial intelligence. So let's dive in and explore the story of us, how our brains came to be, what makes us unique, and what it might take to build a Rosie of our own. Chapter 1. The World Before Brains. Let's rewind the clock way back. Life on Earth existed for over 3 billion years before the brain ever appeared. That's right, brains are a relatively recent development, showing up in the last 15% of life's story. But intelligence, that's been around much longer. To understand how and why brains evolved, we first need to explore the evolution of intelligence itself. About 4 billion years ago, in the volcanic oceans of a lifeless Earth, something extraordinary happened. Amid the chaos of boiling water and volcanic activity, the right combination of molecules came together to form DNA-like chains. These molecules weren't alive, but they had one critical ability. They could replicate themselves. This was the first rebellion against entropy, the universal tendency toward disorder. These self-replicating molecules didn't need to survive individually. As long as they made copies of themselves, they persisted. And with that, the process of evolution began. The next big leap came when these molecules were encased in protective lipid bubbles, forming the first cells. Then ribosomes, kind of molecular factory, emerged, translating DNA into proteins. Proteins were a game changer. Unlike DNA, which is relatively inert, proteins could perform complex tasks. They acted like tiny machines with moving parts that could sense and respond to the environment. Even the simplest single-celled organisms like bacteria had proteins that allowed them to move and perceive their surroundings. These early cells could swim toward favorable conditions and away from danger, exhibiting a primitive form of intelligence. The rise of photosynthesis and the Great Oxygenation Event. For billions of years, life on Earth was dominated by single-celled organisms. Energy was scarce and life struggled to survive. Then about 2.4 billion years ago, a species of bacteria called cyanobacteria discovered photosynthesis. By harnessing sunlight to produce energy, cyanobacteria transformed life on Earth. They created an abundance of energy and in the process released oxygen as a byproduct. This led to the Great Oxygenation Event, which dramatically altered Earth's atmosphere. While oxygen was toxic to many early life forms, it also opened the door to new possibilities. Some microbes evolved to use oxygen for energy through cellular respiration, setting the stage for more complex life. With the advent of cellular respiration, life entered a new phase. Respiratory organisms needed to hunt for sugar, which meant actively pursuing and consuming other life forms. This marked the beginning of an evolutionary arms race. Predators developed new ways to hunt, while prey evolved defenses to survive. This constant back and forth drove the diversification of life and led to the emergence of eukaryotes, cells that were larger, more complex, and far more energy efficient than their bacterial ancestors. Eukaryotes were the first to evolve phagotrophy, the ability to engulf and digest other cells. They also gave rise to multicellular organisms, including plants, fungi, and animals. While plants returned to photosynthesis, fungi and animals continued to rely on respiration, with animals taking on the role of active hunters. The birth of neurons. As animals evolved, they faced a unique challenge. Hunting required speed and precision. This is where neurons came in. Neurons are specialized cells that allow animals to sense and respond to their environments quickly. Unlike plants and fungi, which move slowly and imprecisely, animals needed fast, coordinated movements to catch prey. Neurons made this possible. What's fascinating is that neurons are remarkably similar across all species. The neurons in your brain work the same way as those in a jellyfish. This suggests that neurons evolved once in a common ancestor of all animals and have remained largely unchanged over 600 million years. What sets different species apart isn't the neurons themselves, but how they're wired together. Fungi and animals share a lot in common. They both breathe oxygen, digest food, and descended from a common ancestor. But while animals evolved neurons, fungi didn't. Why? It comes down to their survival strategies. Fungi rely on external digestion, waiting for life to die before breaking it down. Animals, on the other hand, actively hunt and kill their prey. This strategy required fast, specific reflexes, which neurons made possible. In fact, the earliest neurons may have evolved for the simple task of swallowing prey. Edgar Adrian and the universal features of neurons. Fast forward to the 20th century, when scientists like Edgar Adrian began unraveling the mysteries of neurons. Adrian made 3 groundbreaking discoveries. 1. All-or-nothing spikes. Neurons don't send continuous signals. Instead, they fire in bursts, like Morse code. A neuron is either on or off. There is no in-between. 2. Rate coding. The strength of a stimulus, like the weight of an object or the brightness of light, is encoded in the rate at which neurons fire. The more intense the stimulus, the faster the neuron fires. And 3. Adaptation. Neurons adjust their sensitivity based on the environment. This allows them to encode a wide range of stimuli, from the faintest touch to the brightest light, despite their limited firing range. These features, spikes, rate coding, and adaptation, are universal across all animals from jellyfish to humans. They allowed early animals to respond to their environment with speed and precision, giving them a crucial advantage in the evolutionary arms race. The first neural circuits. The earliest animals, like coral polyps, didn't have brains. Instead, they had nerve nets, simple networks of neurons that controlled basic reflexes. For example, when a coral polyp senses food, its neurons trigger a coordinated response to grab and swallow the prey. This required a basic form of logic. Do this, not that. Inhibitory neurons, which suppress certain signals, made this possible. These simple circuits were the first glimmers of intelligence in multicellular life. And so the stage was set. With neurons in place and the evolutionary arms race in full swing, it was only a matter of time before nerve nets evolved into brains. But as we'll see, the story of brains doesn't begin the way you might expect. Chapter 2. The Birth of Good and Bad. Let's take a journey back 600 million years to the dawn of something extraordinary, the first bilaterians. These were the ancestors of almost all animals alive today, including us. They were simple worm-like creatures, but they carried with them the seeds of something revolutionary, brains. And with those brains came the ability to navigate the world by steering, a breakthrough that would forever change the course of evolution. The rise of bilateral symmetry. At first glance, the animal kingdom seems incredibly diverse: ants, alligators, bees, baboons, and so on. But if you look closer, you'll notice something remarkable. Most animals share the same basic body plan. They have a front, with a mouth, brain, and sensory organs, and a back, where waste exits. This bilateral symmetry is a stark contrast to the radial symmetry of our distant cousins, like jellyfish and corals, which have no distinct front or back. Why did bilateral symmetry evolve? It comes down to movement. Radially symmetric animals, like corals, are stationary or drift passively, waiting for food to come to them. But bilateral symmetry is perfect for active navigation. Instead of needing to move in all directions, bilaterians could focus on moving forward and turning left or right. It's a simple, efficient design, so efficient, in fact, that humans have adopted it for cars, planes, and boats. But bilateral symmetry didn't just make movement easier, it also paved the way for something even more important, the first brains. Steering, the first breakthrough. The first bilaterians were tiny worm-like creatures, likely no bigger than a grain of rice. They lived in the shallow, gooey microbial mats of the seafloor, navigating a world filled with food, predators, and environmental challenges. To survive, they needed a way to move toward good things like food and away from bad things like predators or harmful environments. This is where steering came in. Take the modern nematode, C. elegans, a close relative of these ancient bilaterians. With just 302 neurons, it has a brain so simple that we can use it as a window into the past. Despite its simplicity, the nematode displays sophisticated behavior. For example, if you place a nematode on one side of a petri dish and food on the other, it will find the food. It doesn't move randomly. It uses smell to steer. Its strategy is simple but effective. 1. If the smell of food increases, keep moving forward. If the smell of food decreases, turn. This elegant system allows the nematode to navigate without needing a detailed understanding of its environment. It's not thinking about where food is, it's simply following the gradient of the smell. The same principle applies to avoiding danger. Nematodes steer away from light, heat, and harmful chemicals, using sensory neurons to detect these threats and adjust their movements accordingly. The first brains, steering on a multicellular scale. Steering wasn't new. Single-celled organisms like bacteria had been doing it for billions of years, but bacteria use simple chemical processes to change direction, which works well for single cells. For multicellular organisms, steering required a more complex system, neurons. Neurons allowed stimuli like smells or light to trigger coordinated muscle movements, enabling larger organisms to steer effectively. The first brains weren't much more than steering centers. They integrated sensory input and made decisions about which direction to move. For example, nematodes have sensory neurons that detect good and bad stimuli. Positive valence neurons respond to things like food smells, while negative valence neurons respond to dangers like heat or predators. These neurons don't just detect stimuli, they cast votes for whether the nematode should move forward or turn. The brain tallies these votes and makes a single decision, go forward or turn away. The ability to integrate sensory input was a game changer. It allowed bilaterians to navigate complex environments, balancing competing stimuli to make trade-offs. For example, if a nematode smells food on the other side of a dangerous copper barrier, it has to decide whether the reward is worth the risk. The stronger the food smell, the more likely it is to cross the barrier. This kind of decision-making required a centralized brain, something a simple nerve net like those in corals couldn't handle. Valence, the birth of good and bad. To steer effectively, bilaterians needed to categorize the world into good and bad. This is what psychologists and neuroscientists call valence. Valence isn't about morality, it's a primitive evaluation of whether something should be approached or avoided. For nematodes, food smells have positive valence, while predators and harmful chemicals have negative valence. Interestingly, valence isn't fixed. It depends on an animal's internal state. For example, a nematode's response to food smells changes depending on how hungry it is. If it's full, it will ignore the food. If it's hungry, it will steer toward the food, even if there's some risk involved. The ability to adjust valence based on internal states is a hallmark of even the simplest brains. The Roomba and the first brains. The evolution of steering in bilaterians has an interesting parallel in modern robotics. In the 1990s, roboticist Rodney Brooks argued that AI should start with simple behaviors like sensing and moving before tackling complex tasks like reasoning and language. He believed that understanding basic intelligence like steering was the key to building more advanced systems. Excuse me. Brooks' company, iRobot, created the Roomba, a vacuum cleaning robot that navigates using simple rules. The Roomba doesn't map its environment or recognize objects. Instead, it moves randomly, steering away from obstacles and toward its charging station when its battery is low. In many ways, the Roomba's navigation strategy mirrors that of the first bilaterians. Both systems use simple rules to navigate the complex environments without truly understanding them. One of the most impressive features of early brains was their ability to make trade-offs. For example, if a nematode encounters both food and danger, it has to weigh the risks and rewards. This requires integrating inputs from multiple sensory neurons and making a single decision. The brain acts as a central hub, tallying votes from different neurons and choosing the best course of action. This ability to make trade-offs was built on earlier evolutionary innovations. For example, inhibitory neurons, which evolved in coral-like animals to control reflexes, were repurposed in bilaterians to help with decision-making. These neurons allowed bilaterians to choose between mutually exclusive actions, like going forward or turning. The role of internal states. Internal states, like hunger, play a crucial role in decision-making. A nematode's willingness to cross a dangerous barrier depends not only on the external stimuli, food, smell versus danger, but also on its internal state, hunger versus fullness. This flexibility is what makes steering so effective. It allows animals to adapt their behavior to changing circumstances, increasing their chances of survival. Even the Roomba has a rudimentary internal state. When its battery is full, it ignores the signal from its charging station. But when the battery is low, the charging station becomes a high-valence stimulus, and the Roomba steers toward it. Steering required 4 key innovations: a bilateral body plan, valence neurons, a brain to integrate input, and the ability to modulate valence based on internal states. But evolution didn't stop there. The next big leap was the emergence of something even more powerful: emotion. This early kernel of emotion would further enhance the effectiveness of steering, setting the stage for the next chapter in the story of brains. Chapter 3, The Origin of Emotion. Let's talk about emotions. You know, the blood-boiling fury when someone defends a political gaffe you can't stand, or the tingly serenity of lying on a warm beach. Emotions are complex, hard to define, and deeply personal. But at their core, they boil down to 2 universal dimensions: valence, good and bad, and arousal, high or low energy. These 2 features, collectively called affect, are the ancient foundation of all emotions, shared not just across human cultures, but throughout the animal kingdom. And believe it or not, they evolved for one surprisingly mundane purpose: steering. The simplicity of affect. Affect is universal. Whether you're calm, elated, tense, or upset, your emotional state can be mapped onto a grid of valence and arousal. For example, lying on a beach might be a low arousal, positive valence state, while riding a roller coaster, if you enjoy it, is high arousal and positive valence. Similarly, depression is a low arousal event and negative valence, while rage is a high arousal, negative valence. This universality isn't just a human thing. Across cultures, people have words for valence and arousal, and even newborns express these states through crying or smiling. But it doesn't stop there. Affect is found throughout the animal kingdom. Even simple creatures like nematodes, tiny worms, affect primitive forms of affect. So why did affect evolve in the first place? Steering in the dark. Why affect evolved? Imagine a hungry nematode in a petri dish. It doesn't just sit there waiting for food to magically appear. Instead, it swims around searching for food. When it stumbles upon a hidden patch of food, it slows down and starts turning more frequently, staying in the area to exploit the resource. This shift from escaping, rapid swimming, to exploiting, local searching, is a basic form of affect. Negative valence states, like hunger, trigger escape behaviors, while positive valence states, like finding food, trigger exploitation. These affective states persist even after the initial stimulus is gone. A nematode doesn't stop searching for food the moment it stops smelling it. This persistence is crucial because sensory stimuli in the real world are often fleeting. A passing whiff of food or a brief hint of a predator might be the only clue a nematode gets. By maintaining an effective state, the nematode can continue steering toward food or away from danger, even in the absence of direct sensory input. It's like flying a plane through a fog, relying on the brief flickers of information to guide your path. Even modern robots like Roomba use a similar strategy. When Roomba detects a patch of dirt, it doesn't just clean the spot and move on. Instead, it switches to a local search mode, circling the area to find more dirt. This persistence, triggered by a single detection, mirrors the effective states of nematodes. It's a simple but effective way to navigate a complex world. Dopamine and serotonin, the chemicals of affect. The brains of nematodes use chemicals called neuromodulators to generate affective states. Two of the most famous neuromodulators are dopamine and serotonin, which play distinct roles in steering behavior. Dopamine is released when a nematode detects food nearby. Dopamine triggers the state of exploitation. It's the "something good is nearby" chemical, driving the nematode to search its surrounding for food. Serotonin, released when food is actually consumed. Serotonin triggers the state of satiation. It's the "something good is happening" chemical, signaling the nematode to stop searching and enjoy the reward. The basic dichotomy between dopamine and serotonin has been conserved across evolution. In humans, dopamine is released when we anticipate rewards, like seeing a delicious meal or the finish line of a race, while serotonin is released when we experience those rewards, like eating the meal or crossing the finish line. Dopamine drives wanting, while serotonin drives satisfaction. But dopamine isn't the pleasure chemical it's often made out to be. Experiments with rats have shown that increasing dopamine levels make them more motivated to pursue rewards, but it doesn't make them enjoy those rewards anymore. In fact, rats with depleted dopamine still enjoy food. They just lack the motivation to seek it out. Dopamine is about wanting, not liking. Stress and the evolution of negative affect. Affective states aren't just about pursuing rewards, they're also about avoiding threats. Negative valence states like fear or stress are triggered by neuromodulators like adrenaline and norepinephrine. These chemicals activate the fight or flight response, increasing heart rate, suppressing digestion, and diverting energy to muscles for rapid escape. But stress is costly. It consumes a lot of energy, so evolution came up with a counterbalance: anti-stress chemicals like opioids. After a stressful event, opioids help the body recover by turning off the stress response and restoring functions like digestion and reproduction. They also enhance serotonin and dopamine signals, making recovery feel good. This is why opioids are such powerful painkillers. They increase pleasure and reduce pain. However, prolonged stress can lead to a different state: chronic stress. When a nematode is exposed to an inescapable stressor, like extreme heat or toxic chemicals, it eventually gives up. It stops trying to escape and enters a low arousal, negative valence state. This is the evolutionary precursor to depression. Chronic stress shuts down arousal and motivation, conserving energy in the face of inescapable challenges. In humans, this manifests as anhedonia, the inability to feel pleasure. The ancient roots of emotion. The basic template of affect, valence, and arousal evolved over 550 million years ago in early bilaterians. These primitive affect states were simple answers to two fundamental questions. 1. Do I want to expend energy moving or not? Arousal. 2. Do I want to stay here or leave? Valence. Neuromodulators like dopamine and serotonin enforce these answers, modulating behavior to maximize survival. Over time, these basic mechanisms were elaborated into the complex emotions we experience today. But at their core, our emotions are still built on the scaffolding of these ancient affective states. So the next time you feel a rush of excitement or a pang of sadness, remember, you're tapping into a system that's been evolving for over 500 million years, and it all started with the simple need to steer. Chapter 4. Associating, Predicting, and the Dawn of Learning. Let's dive into the story of how learning began. It's a tale that starts with a Russian physiologist, Ivan Pavlov, and his famous dogs, but it stretches back over 550 million years to the first bilaterians. Learning, as we know it, didn't emerge as a grand evolutionary leap. Instead, it began as a simple trick to help early animals steer better. From that humble beginning, it grew into the foundation of everything our brains do today. Pavlov and the discovery of associative learning. In 1904, Ivan Pavlov stood before an audience in Stockholm, fresh off winning the Nobel Prize, not for psychology as many assume, but for his groundbreaking work in physiology. Pavlov had revolutionized the study of the digestive system, developing techniques to measure the activity of organs like the salivary glands in live, healthy dogs. But during his experiments, he noticed something peculiar. His dogs would start salivating before they even tasted food, simply at the sight or sound of something they associated with food. Pavlov initially saw this as an annoying psychic stimulation that interfered with his measurements. But over time, he realized it was something far more profound. Pavlov discovered that dogs could form conditional reflexes. For example, if a buzzer sounded before food was presented, the dogs would eventually salivate at the sound of the buzzer alone. This was different from unconditional reflexes like salivating when food touches the tongue, which are innate and require no learning. Pavlov's work revealed that animals could learn associations between stimuli, and this learning happened automatically without conscious effort. Learning without a brain. Pavlov's findings hinted at something remarkable. Learning didn't require a complex brain. Even a rat with its brain removed could form simple associations using just the circuits in its spinal cord. This suggested that associative learning was an ancient evolutionary innovation, one that likely emerged with the first bilaterians. Take nematodes, for example. These tiny worms can learn to associate salt with hunger. If you expose nematodes to salty water without food, they'll later avoid salt, even though they normally find it attractive. Similarly, slugs can learn to associate a gentle tap with an electric shock, withdrawing at the tap alone after repeated pairings. This ability to tweak the goodness or the badness of things based on experience is a hallmark of associative learning, and it's found across all bilaterians. But it's absent in our radially symmetric cousins, like jellyfish and corals, which lack the need for such learning. After all, a coral polyp doesn't need to remember anything. It just sits in place, waiting for food to drift by. The evolutionary pressure to learn. For early bilaterians, learning was a matter of survival. These animals didn't just sit still, they moved through their environment, steering toward food and away from danger. But the world is unpredictable. A chemical that signals food in one environment might signal predators in another. The ability to adjust steering decisions based on experience gave bilaterians a huge evolutionary advantage. Associative learning allowed them to remember which cues led to rewards or punishments and adapt their behavior accordingly. The mechanics of learning: acquisition, extinction, and recovery. Pavlov's experience didn't just reveal that animals could learn associations, they also showed how these associations could change over time. For example, acquisition. The more times a buzzer was paired with food, the stronger the association became, and the more the dog salivated at the sound of the buzzer. Extinction. If the buzzer was repeatedly sounded without food, the dogs eventually stopped salivating. The association faded. Spontaneous recovery. Even after extinction, the association wasn't truly gone. If the buzzer was sounded again in a few days, the dogs would start salivating again, as if the memory had resurfaced. And reacquisition. If the buzzer was paired with food again, the dogs relearned the association much faster than the first time. These processes, acquisition, extinction, spontaneous recovery, and reacquisition, allowed early bilaterians to navigate a constantly changing world. For example, if a worm found food near salt, but later encountered salt without food, it might stop associating salt with food. But if food reappeared near the salt, the worm would quickly relearn the association, ensuring it didn't miss out on future meals. Solving the credit assignment problem. Associative learning comes with a challenge. When something good or bad happens, how does the brain know which cue to associate with it? This is known as the credit assignment problem. Early bilaterians developed 4 clever tricks to solve it. 1. Eligibility traces. Associations were only formed if the predictive cue occurred within a short time window before the event, such as a tap followed by a shock within 1 second. This ensured that only relevant cues were linked to outcomes. 2. Overshadowing. When multiple cues were present, the strongest one, such as a bright light over a faint smell, was used as the predictor. 3. Latent inhibition. Familiar stimuli were treated as background noise and ignored. For example, if a slug had experienced a certain texture many times without consequence, it wouldn't associate that texture with a shock. And 4. Blocking. Once an association was formed, new cues that overlapped with the original cue were ignored. If a slug had already learned that a tap predicted a shock, it wouldn't associate a new smell with the shock. These tricks weren't perfect, but they were good enough to help early bilaterians navigate their environment and make better decisions. The physical basis of learning, synaptic plasticity. For centuries, philosophers speculated about how learning worked. Plato imagined the brain as a wax tablet where experiences left impressions. Descartes thought memories were folds in the brain, but the real breakthrough came in the 20th century with the discovery of neurons and synapses. Learning, it turns out, happens at the synapses, the connections between the neurons. When two neurons fire together, their connection strengthens, making it easier for one to activate the other in the future. This process, known as Hebbian learning, or neurons fire together, wire together, is the basis of associative learning. For example, if a sensory neuron detects a tap and a motor neuron triggers a withdrawal reflex, their connection strengthens if the tap is consistently followed by a shock. Over time, the tap alone can trigger the reflex. Neuromodulators like dopamine and serotonin play a key role in this process. Dopamine, for instance, signals the presence of a reward, enhancing the strength of relevant synapses. This allows animals to learn which cues predict good things, like food. The legacy of associative learning. The ability to form associations was a game changer for early bilaterians. It allowed them to adapt to their environments, remember past experiences, and make better decisions. While their learning abilities were limited, they couldn't recognize objects, predict timing, or understand complex patterns. They laid the foundation for all future learning. The same mechanism that helped nematodes tweak their steering are still at work in our brains today, enabling us to learn languages, play music, and navigate the world. Learning wasn't the primary function of the first brains. It was just a feature, a tool to optimize steering. But over time, it became the brain's central purpose. And as we'll see in the next chapter, the evolution of learning didn't stop with simple associations. It paved the way for even more sophisticated forms of intelligence, transforming the brain into the incredible organ we know today. Chapter 5. The Cambrian Explosion and the Birth of Reinforcement Learning. Let's take a leap forward in time, about 50 million years after the first bilaterians wiggled their way into existence. Welcome to the Cambrian period, a time of extraordinary change and innovation in the history of life on Earth. This was the era of the Cambrian explosion, a period when life diversified at an unprecedented rate, and the evolutionary arms race between predators and prey reached new heights. It was also the time when our ancestors, the first vertebrates, made their debut, bringing with them a revolutionary new brain capable of something remarkable: reinforcement learning. The Cambrian world, a zoo of terrifying creatures. The Cambrian period, which spanned from 540 to 485 million years ago, was a world unlike anything that came before. Gone were the gooey microbial mats and slow-moving creatures, and in their place was a bustling, chaotic ecosystem dominated by massive arthropods, ancestors of insects, spiders, and crustaceans. These creatures were armed with oversized claws, armored shells, and a terrifying presence. Some grew to over 5 feet long, making them the apex predators of their time. Amid this Cambrian cacophony, our ancestors were far less conspicuous. They were small, fish-like creatures only a few inches long, with fins, gills, and a spinal cord. But these unassuming vertebrates carried with them the seeds of something extraordinary, a new kind of brain. The vertebrate brain, a new template for intelligence. The brains of early vertebrates were a game changer. Unlike the simple decentralized nervous systems of invertebrates like nematodes or arthropods, vertebrate brains were organized into distinct regions with specialized functions. This template, forged during the Cambrian explosion, is still present in all vertebrates today, from fish to humans. The vertebrate brain develops in 3 primary stages, forming 3 main regions: the forebrain, midbrain, and hindbrain. The forebrain further splits into 2 subsystems, one that becomes the cortex and basal ganglia, and another becomes the thalamus and hypothalamus. These 6 structures, cortex, basal ganglia, thalamus, hypothalamus, midbrain, and hindbrain, are shared across all vertebrates. Even the brain of a jawless lamprey fish, one of our most distant vertebrate relatives, share these same structures. This new brain architecture allowed vertebrates to process information in more sophisticated ways, laying the groundwork for a breakthrough in learning, reinforcement learning. Thorndike's puzzle boxes, the discovery of reinforcement learning. Fast forward to the late 19th century, when an American psychologist named Edward Thorndike was studying how animals learn. Thorndike, a shy but brilliant researcher, wanted to understand the principles of learning, not just in humans, but across all animals. His experiments with chickens, cats, and dogs led to a groundbreaking discovery. Animals learn through trial and error. Thorndike's most famous experiments involved puzzle boxes. He placed animals in cages with simple mechanisms like latches, buttons, or hoops that, when manipulated, would open the door and allow the animal to escape. At first, the animals tried a variety of random behaviors, scratching, pacing, howling. Eventually, they stumbled upon the correct action, escaped, and received a reward, usually food. Over repeated trials, the animals became faster and more efficient, immediately performing the correct action to escape. Thorndike called this process trial and error learning and summarized it in his famous law of effect. Behaviors that lead to satisfying outcomes are more likely to be repeated, while behaviors that lead to discomfort are less likely to occur. In modern terms, we call this reinforcement learning. Positive outcomes reinforce behaviors, while negative outcomes discourage them. The surprising smarts of fish. Thorndike's experiments weren't limited to cats and dogs. He also tested fish, and the results were just as impressive. In one experiment, Thorndike placed a fish in a tank with transparent walls that had hidden openings. The fish, motivated to escape the brightly lit side of the tank, initially swam around randomly, bumping into the walls. Eventually, it found the openings and made its way to the darker, more desirable side. With repeated trials, the fish learned to navigate the maze quickly and efficiently. This ability to learn arbitrary sequences of actions through trial and error is a hallmark of reinforcement learning. Fish can learn to push buttons, swim through escape hatches, and even jump through hoops to get food. They can remember these tasks for months or even years. This level of learning is far beyond the capability of simpler bilaterians like nematodes or slugs, which cannot be trained to perform complex sequences of action. Why reinforcement learning was a breakthrough? Reinforcement learning might seem simple. Just reinforce good behaviors and punish bad ones, but it's actually a profound evolutionary innovation. Early bilaterians, like nematodes, lacked the neural machinery to learn through trial and error. Their brains were too simple, limited to basic associative learning, such as pairing a stimulus with a response. Vertebrate brains, on the other hand, had the complexity needed to evaluate sequences of actions, assign credit to specific behaviors, and adjust future actions accordingly. This ability to learn from experience gave vertebrates a massive advantage in the Cambrian arms race. A fish that could learn to avoid predators, find food, or navigate complex environments was far more likely to survive and reproduce than one that couldn't. So what are the challenges of reinforcement learning? Reinforcement learning isn't as easy as it sounds. When an animal receives a reward or a punishment, how does its brain know which action to reinforce or discourage? This is known as the credit assignment problem we discussed earlier. Early vertebrate brains solved this problem using a combination of clever tricks. 1. Trial and error. Animals performed random actions and gradually refined their behavior based on outcomes. 2. Reinforcement and punishment. Positive outcomes reinforce behaviors while negative outcomes discourage them. And 3. Memory. Vertebrate brains could store information about past experiences, allowing animals to learn from their mistakes and successes. These mechanisms, though simple, were incredibly effective. They allowed vertebrates to adapt to their environments, learn complex behaviors, and outcompete other animals. The legacy of reinforcement learning. Reinforcement learning was the second major breakthrough in brain evolution, following the development of steering in early bilaterians. It transformed the vertebrate brain into a powerful learning machine capable of adapting to new challenges and opportunities. This ability to learn arbitrary sequences of actions through trial and error laid the foundation for more advanced forms of learning, which we'll explore in the chapters to come. So the next time you see a fish swimming in a tank, remember it's not just a mindless creature, it's the descendant of an ancient lineage that pioneered one of the most important innovations in the history of brains. And it all started 500 million years ago in the chaotic, predator-filled world of the Cambrian explosion. Chapter 6, The Evolution of Temporal Difference Learning. let's talk about the most fascinating breakthroughs in brain evolution. The emergence of temporal difference learning, TD. This is the story of how vertebrate brains became capable of solving one of the most challenging problems in reinforcement learning, assigning credit to actions that lead to rewards or punishments, even when those outcomes are delayed. It's a tale that spans 500 million years, from the first vertebrates to modern AI systems, and it all starts with a problem that stumped early researchers in artificial intelligence: the temporal credit assignment problem. In the 1950s, Marvin Minsky, a doctoral student at Princeton, built one of the first reinforcement learning algorithms, the Stochastic Neural Analog Reinforcement Calculator, SNARC, inspired by Thorndike's law of effect. Minsky's algorithm used trial and error learning to navigate mazes. It worked by strengthening the connections or synapses that were active when the system successfully escaped a maze. But there was a problem. SNARC struggled with anything more complex than a simple maze. The issue was the temporal credit assignment problem. Imagine teaching an AI to play checkers. If the AI wins a game, which of the hundreds of moves should get credit for the victory? If it loses, which moves should be blamed? Reinforcing only the most recent moves ignores the importance of earlier decisions, while reinforcing all moves equally dilutes the impact of critical actions. Minsky realized that without a way to assign credit across time, reinforcement learning would fail in complex scenarios. A breakthrough in AI, temporal difference learning. Decades later, in 1984, Richard Sutton proposed a solution in his PhD dissertation, temporal difference learning. Sutton's idea was simple but revolutionary. Instead of reinforcing actions based on actual rewards, why not reinforce them based on predicted rewards? He introduced the concept of an actor and a critic. The critic predicts the likelihood of future rewards at every moment, while the actor chooses actions and gets rewarded whenever the critic's prediction improves. This way, the system learns not just from the final outcome, but from incremental changes in its expectations. For example, in a game of checkers, if a clever move improves the AI's position, the critic signals this improvement and the actor reinforces the move, even if the game isn't over yet. This approach allows the system to learn as it goes, solving the temporal credit assignment problem. Sutton's simulations showed that this method worked, but it wasn't until Gerald Tesoro applied it to backgammon in the 1990s that its potential was fully realized. Tesoro's TD Gammon, which learned to play backgammon using temporal difference learning, became one of the best backgammon players in the world, outperforming even expert humans. This success demonstrated that TD learning wasn't just a clever idea, it was a powerful tool for solving real-world problems. The biological connection, dopamine and TD learning. While Sutton's work revolutionized AI, he suspected that TD learning wasn't just a technological invention, it might also reflect how animal brains solve the temporal credit assignment problem. This idea gained traction in the 1990s when Peter Dayan and Reed Montague connected Sutton's theory to the brain's dopamine system. Dopamine, a neuromodulator found in all vertebrates, has long been associated with reinforcement. Rats, for example, will press a lever thousands of times an hour if it stimulates their dopamine neurons, even ignoring food to the point of starvation. What exactly was dopamine signaling? Early theories suggested it was a pleasure chemical, but experiments by Wolfram Schultz revealed a more nuanced role. Schultz showed that dopamine neurons initially respond to rewards, like sugar water for monkeys, but after a few trials, the dopamine response shifts to the predictive cue, a picture signaling the reward, and disappears when the reward itself is delivered. If the expected reward is omitted, dopamine activity decreases, signaling disappointment. This pattern matched Sutton's temporal difference learning signal perfectly. Dopamine encodes changes in predicted future rewards, not the rewards themselves. For example, positive temporal difference. A cue predicts a reward, increasing dopamine activity. No temporal difference. The reward is delivered as expected and dopamine remains steady. Negative temporal difference. As expected, an expected reward is omitted, decreasing dopamine activity. This discovery revealed that dopamine neurons implement a biological version of TD learning, allowing vertebrates to solve the temporal credit assignment problem. The role of basal ganglia. At the heart of this system is the basal ganglia, a brain structure found in all vertebrates. The basal ganglia acts as a global puppeteer of behavior, gating and ungating actions based on input from the cortex, thalamus, and midbrain. It also receives input from dopamine neurons, which modulate its activity. Here's how it works. 1. The basal ganglia monitors an animal's action and environment. 2. Dopamine signals from the midbrain reinforce or punish specific actions. And 3. Over time, the basal ganglia learns to repeat actions that maximize dopamine release and inhibit actions that reduce it. This system is remarkably conserved across vertebrates. The basal ganglia of a human is nearly identical to that of lamprey fish, a species whose ancestors were among the first vertebrates 500 million years ago. This suggests that the basal ganglia's role in reinforcement learning has been a core feature of vertebrate brains since their inception. Disappointment, relief, and the perception of time. TD learning also gave rise to two familiar emotions, disappointment and relief. These emotions are emergent properties of a brain designed to predict future rewards. For example, disappointment occurs when an expected reward is omitted, triggering a negative temporal difference signal. Relief occurs when an expected punishment is avoided, triggering a positive temporal difference signal. These mechanisms allow vertebrates to learn not only from rewards and punishments, but also from their absence. For instance, a fish trained to avoid a zap by swimming to the other side of the tank learns to associate the omission of the zap with its actions. This type of learning, called an avoidance task, is unique to vertebrates and relies on the precise timing enabled by TD learning. Timing is another critical feature of vertebrate brains. Unlike simpler bilaterians, which can't associate events separated by more than a few seconds, vertebrates can learn precise time intervals. For example, a fish can learn that a zap occurs exactly 5 seconds after a light turns on and adjust its behavior accordingly. This ability to track time is essential for TD learning, as it allows the brain to predict when rewards or punishments should occur. The hypothalamus and the actor-critic system. The hypothalamus is another key structure in vertebrate brains, playing a complementary role to the basal ganglia. It acts as the judge of actual rewards and punishments, triggering dopamine release when an animal's needs are met, such as food when hungry. The basal ganglia, in turn, uses this feedback to learn which actions to repeat. Over time, the basal ganglia develops its own internal critic, capable of predicting future rewards and triggering dopamine release independently of the hypothalamus. This internal actor-critic system is the biological manifestation of Sutton's TD learning model, allowing vertebrates to learn from predicted rewards rather than actual ones. The legacy of TD learning. The emergence of TD learning in vertebrate brains was a transformative moment in evolution. It allowed animals to solve the temporal credit assignment problem, learn from delayed outcomes, and adapt their behavior in complex dynamic environments. This capability laid the foundation for more advanced forms of learning and intelligence, from the ability to plan for the future to the development of emotions like disappointments and relief. Today, TD learning continues to shape our understanding of both biology and technology. It bridges the gap between neuroscience and AI, showing how ancient evolutionary innovations can inspire modern breakthroughs. And it all started 500 million years ago with the first vertebrates and their remarkable brains. Chapter 7. The Problems of Pattern Recognition. Let's dive into one of the most fascinating breakthroughs in brain evolution, the ability to recognize patterns. This chapter takes us back 500 million years to the Cambrian period, where our fish-like ancestor swam unknowingly into danger. It's a story of survival, learning, and the emergence of a skill that would forever change the trajectory of life on Earth. Pattern recognition. A Cambrian tale, the birth of pattern recognition. Picture this: our tiny vertebrate ancestor, an inch-long fish-like creature, is swimming through a Cambrian ocean, hunting for coral larvae. Unbeknownst to her, she is being hunted by a terrifying arthropod with spiked claws. As the predator lunges, her reflexes kick in. She detects the movement, floods her body with norepinephrine, and escapes just in time. But this time, something remarkable happens. She remembers. She remembers the smell of the predator, the sight of its eyes peeking through the sand. The next time she encounters these cues, she'll know how to avoid them. This is the birth of pattern recognition. The challenge of recognizing patterns. Recognizing patterns, whether it's a smell, a sound, or a sight, might seem effortless to us, but it's an incredibly complex task. Early bilaterians, like our nematode-like ancestors, couldn't do this. Their perception of the world was limited to individual sensory neurons. For example, a single photosensitive neuron could detect light, or a mechanosensory neuron could detect touch. While this was enough for basic steering, it provided a very limited understanding of the world. In contrast, early vertebrates developed the ability to recognize patterns across multiple neurons. For example, smells are not single molecules, but a combination of many. When you smell pulled pork, your brain isn't detecting a pulled pork molecule. There's no such thing. Instead, it's recognizing a unique pattern of activated olfactory neurons. This ability to decode patterns dramatically expanded what animals could perceive, allowing them to recognize a vast array of smells, sights, and sounds. The two big problems of pattern recognition. Pattern recognition isn't just hard, it's really hard. Vertebrate brains had to solve two major challenges. One, the discrimination problem. How do you distinguish between overlapping patterns? For example, the smell of a predator might overlap with the smell of food or a mate. The brain needs to recognize these as distinct patterns and respond appropriately. And two, the generalization problem. How do you recognize a pattern that's similar but not identical? For instance, the smell of a predator might vary slightly depending on its age, diet, or environment. The brain needs to generalize from past experiences to recognize new variations of the same pattern. How vertebrate brains solve these problems. The vertebrate brain tackled these challenges using the cortex, a structure that had first appeared in early vertebrates. The cortex is home to pyramidal neurons, which are uniquely suited for this pattern recognition. These neurons receive input from thousands of synapses, allowing them to process complex patterns. Two key features of the cortex help solve the discrimination problem. One, dimensionality expansion. A small number of sensory neurons connect to a much larger number of cortical neurons, creating a more detailed representation of sensory input. And two, sparse connections. Each sensory neuron connects to only a subset of cortical neurons, ensuring that overlapping patterns are separated in the cortex. To solve the generalization problems, the cortex uses a trick called autoassociation. When a pattern activates a group of pyramidal neurons, these neurons wire themselves together through Hebbian plasticity, the neurons that fire together wire together principle. This means the next time a similar pattern appears, the entire group of neurons is reactivated, allowing the brain to recognize the pattern, even if it's incomplete or slightly different. Pattern recognition in AI, lessons from the brain. Modern AI systems, like the facial recognition software on your phone, face the same challenges of discrimination and generalization. They solve these problems using artificial neural networks, which are inspired by the brain. For example, convolutional neural networks, CNNs, mimic the hierarchical structure of the visual cortex, breaking down images into simple features like edges and lines, and then combining them into more complex objects. However, there are key differences between AI and the brain. AI systems rely on supervised learning, where they're trained with labeled examples, such as, this is a cat, this is a dog. The brain, on the other hand, learns patterns without supervision. Additionally, AI systems struggle with catastrophic forgetting. When they learn new patterns, they often overwrite old ones. Vertebrate brains, even those of fish, avoid this problem remarkably well, retaining memories for years despite constantly learning new things. The invariance problem. Recognizing objects despite variations. One of the most impressive feats of vertebrate brains is their ability to solve the invariance problem, recognizing an object the same despite changes in its appearance. For example, you can recognize a chair whether it's rotated upside down or viewed from a different angle. This is much harder than it seems because the pattern of neurons activated in your retina changes completely depending on the object's orientation. In the 1950s, neuroscientists David Hubble and Torsten Wiesel discovered how the visual cortex solves this problem. They found that neurons in the visual cortex are organized hierarchically. V1 detects simple features like edges and lines. V2 and V4 combine these features into more complex shapes. And IT, in inferotemporal cortex, recognizes whole objects like faces or chairs regardless of their position or orientation. Inspired by this, computer scientist Kunihiko Fukushima developed convolutional neural networks which use a similar hierarchical approach to recognize objects and images. Today, CNNs power many AI systems from self-driving cars to medical imaging software. Fish brains, a mystery of simplicity and sophistication. While CNNs were inspired by the mammalian visual cortex, even the simpler brains of fish can solve the invariance problem. For example, goldfish can recognize a picture of a frog even when it's shown from a new angle they've never seen before. How do fish achieve this with their tiny brains? The answer remains a mystery, but researchers suspect it involves complex interactions between the cortex and the thalamus, a structure at the center of the brain. The coevolution of pattern recognition and sensory organs. The evolution of pattern recognition didn't happen in isolation. As vertebrate brains became better at recognizing patterns, sensory organs like the eyes, ears, and noses became more sophisticated. This coevolution created a feedback loop. Better pattern recognition made more detailed sensory input useful, and more detailed sensory input made better pattern recognition necessary. This also tied into the evolution of reinforcement learning. The more patterns a brain could recognize, the more actions it could learn to take in response. This interplay between pattern recognition, sensory organs, and reinforcement learning laid the foundation for the complex behaviors we see in vertebrates today. The legacy of pattern recognition. The ability to recognize patterns transformed the way animals interact with the world. It allowed vertebrates to perceive their environments in unprecedented detail, learn from their experiences, and adapt to new challenges. From the first fish to modern humans, pattern recognition has been a cornerstone of intelligence, enabling everything from escaping predators to unlocking your iPhone. So the next time you recognize a familiar face or smell, remember, you're using a skill that began 500 million years ago in the brain of a tiny fish swimming through the Cambrian Ocean. This is the end of part 1 of A Brief History of Intelligence by Max Bennett. I think you'll find the insights for this book very revealing because he provides parallels to artificial intelligence and how AI has adapted using cues from how the brain has adapted over time. It's a great book. If you're interested in learning more, pick up a copy. That's where we'll leave it for this week. Thanks for joining. That's a wrap.